Good afternoon, everyone. Thank you for logging here today. I'm Virginie Sinet-Mathiot, and I'm going to talk about biomolecular archaeology, and more specifically, paleoproteomics. When combined with the archaeological analysis, it provides us the possibility to understand Neanderthal behavior through the reconstruction of human diet and subsistence strategies. Paleolithic faunal assemblages are commonly highly fragmented by numerous taphonomic agents, including carnivores and humans. This high level of bone fragmentation prevents proper species identification for most of the bone specimens we excavate. Previous studies of past human behavior have been relying on morphologically identifiable fauna, potentially excluding vast majority of the bone specimens. Thus, bone fragmentation not only leads to a loss of taxonomic identification, but also to a loss of hominin behavioral information, with current interpretation based on a relatively small proportion of identifiable remains. We can therefore ask to what extent this identifiable portion is representative of the whole assemblage. How can we further study the unidentifiable component? Furthermore, the fragmentation of bone remains at archaeological sites can often result from specific human behaviors. So, how does the fragmented fraction of the bone assemblages found on archaeological sites inform us about hunting strategies and subsistence behavior? Recent developments of biomolecular methods allow us to exploit the collagen preserved in bone fragments. Zooarchaeology by mass spectrometry is a minimally destructive proteomic method which focuses on unidentifiable bone fragments. The analysis of collagen protein type 1 provides a taxonomic identification based on a protein amino acid sequence variation. Collagen type 1 presents the advantage of being easily accessible, phylogenetically informative, and survives beyond the temporal range of ancient DNA. This method allows us to finally exploit a large portion of bones preserved in Pleistocene sites. This component of the archaeological bone assemblages have been neglected up to now due to the lack of morphological criteria. Nonetheless, the analysis of these fragments provide a direct link of human behavior and can now participate to the understanding of the subsistence strategies of human groups occupying the site. So how does it work? In short, soluble collagen is extracted by unfolding the molecule. Digestion with trypsin is used to cleave the protein into peptides, which are then analyzed through a mass spectrometer. The taxonomic identification is made through the mass identification of a list of 9 peptide markers in comparison to a database. We obtain spectra, schematically represented here, with reindeer on top and horse at the bottom. And the difference occurs in the changes of amino acid in the peptide sequence, represented here by the black dots in the chains. Up to now, most large-scale zoom screening studies were only looking for hominin remains, which generally represent less than 1% of bone remains found on a site. The other 99% of specimens identified are most often used only to answer ecological questions, trying to improve the faunal spectrum, but are generally not analyzed from a behavioral perspective. Combining the analysis of the bone surface modifications with their biomolecular analysis can provide fresh insights into past human subsistence behavior. So we investigate here the relationship between faunal composition and bone fragmentation. ZooMS provide datasets comparable with zooarchaeology, potentially giving fresh insights into subsistence behavior in relation to faunal carcass processing and selection, but also bringing a more complete picture of assemblage composition. We will present today 2 case studies where we will discuss issues of subsistence and its change through a sequence using complementary zooms and the archaeology datasets. So first case study, Patrokio Cave, which is located in central Bulgaria in the Balkans and is represented here on the slide by the orange star on the map. The site have been first excavated during a campaign between 1970 and 1975, revealing a stratigraphic sequence spanning from the late Middle Paleolithic to the Upper Paleolithic. The excavation allowed the recovery of a large faunal collection. The site is notably known for its distinctive lithic assemblage, the Bachocurian, recently recognized as being part of the initial Upper Paleolithic. Recent work at Bachocur Cave reports new Homo sapiens remains associated with a large dataset of radiocarbon dates, pushing back in time the first dispersal of Upper Paleolithic Homo sapiens across Eurasia, but also the potential interaction with late Neanderthal still present elsewhere in Europe. So in 2015, an international team has started a new excavation campaign at Bachocur Cave, seeking to clarify the stratigraphy and understand human behavior associated with the occupation of the site. Two sectors with similar and well-preserved sequences were excavated, the main sector and the niche one. Both sectors correlate in terms of stratigraphy, as illustrated by the figure on the right of the slide. The radiocarbon chronology provides a secure chronological context for the layers associated with the Mousterian to the Upper Paleolithic. So layer I is dated to approximately 45,000 to 43,000 BP. Layer J is dated to approximately 49,000 to 46,000 BP. And layer K is at the limit for radiocarbon and is older than 41,000 BP. These dates overlap with Heinrich event 5, illustrating changeable climates and environments within this sequence. In order to investigate any differences in the occupation of the site related to the arrival of Homo sapiens, we will focus on these three layers. A large collection of bone tools and personal ornaments have been discovered among the archaeological material, and all associated with the initial Upper Paleolithic. Objects recovered include pendants made from perforated and grooved teeth of mainly cave bear, along with awls, lissoirs, and ivory beads. We can see the pictures of these bone tools on the left of the slide. The human remains, identified using protein mass spectrometry, comprise 6 bone fragments that we can see on the right of the slide, in addition to one tooth. All were recovered from layer I and J and are Homo sapiens with no fossils material yet identified from the Middle Paleolithic layer K. The paleontological and zooarchaeological analysis conducted by Jeff Smith and Rosen Spessoff contribute to the construction of a large identifiable faunal assemblage with more than 7,400 bones studied to date and almost 1,500 identified from layers I, J, and K. The bone preservation is excellent, allowing an identification rate of around 25%, as illustrated by the figure on the left of the slide. And we have a high readability of the bone surfaces to assess the bone modification and factors of accumulation for both initial Upper Paleolithic layers, so I and J, and Middle Paleolithic layer, K. The natural taphonomic agents are limited, with low proportion of high degree of weathering and limited abrasion. The faunal spectrum is dominated by Ursidae and both bison, alongside cervids, equids, and caprines. The presence of woolly mammoth, reindeer, giant deer, and wolverine in layer I and J is indicative of at least one period of extreme cold represented within these deposits. This would correlate with the date ranges provided, showing an overlap with Heinrich event 5. So here we present the number of identified specimens for both the morphology component on the left and the ZooMS analysis on the right. Some of the taxonomic identification based on morphology have been grouped under a broader identification for a matter of standardization, and in order to facilitate also the comparison with the ZooMS component. These comparative tables illustrate well the similarities between both phonospectrum, but most importantly, how complementary is the ZooMS component by providing considerable additional data to the analysis of the bone remains, especially for layers J and K. So more than 2,000 unidentifiable bone and teeth fragments have been sampled and analyzed through ZooMS in order to enlarge our understanding of species distribution among the large portion of fragmented and unidentifiable material, but most importantly, to assess bone fragmentation and any specific human behavior related to it. The phonospectra obtained for layers I, J, and K show similarities with the morphologically identified material, with Ursidae and boss bison as dominant taxon, followed by equids, capra, and cervids. We have here a selection of bone specimens with anthropogenic surface modification that were radiocarbon dated and recently published, as you can see on the left of the slide. When looking at the dominant species, the analysis of bone surface modification show an accumulation of faunal remains mainly driven by human activities, especially increasing from layer K to I, which show a higher taxonomic diversity. This pattern can be observed from the ZooMS and the zooarchaeological analysis. The exploitation of carnivores such as Ursidae, but also Panthera, Lepoids, or even Hyenidae opened the discussion on site occupation, complex interaction with carnivores, and subsistence behavior at Batrocure Cave. The opposite pattern of the anthropogenic modification is observed here. So here is shown the proportion of bones modified by carnivores for the dominant taxon from layer K to I. We seem to have a decrease of bone modifications made by the carnivore from layer K to I, showing a change in the occupation of the site and or the competition and relation with carnivores. This is particularly visible from the ZooMS data, which nicely fill the gap in the morphologically identified assemblage, notably for layer J. So to sum up, layer K is marked by Neanderthal occupation and includes an important proportion of carnivore modifications relatively to anthropogenic marks. This pattern shifts slowly in J, but mainly in layer I, which is clearly accumulated through human subsistence behavior with lower carnivore modifications. These preliminary results obtained from the ZooMS analysis emphasize here a clear shift in site occupation and potential competition with carnivores during Neanderthal occupation. So during the second part of this presentation, we will discuss another aspect of the potential of ZooMS for the study of human evolution through our study at Fumane Cave. So Fumane Cave is located at the bottom of the Venetian Prealps in northern Italy, as illustrated by the orange star on the map. It was first excavated in 1964 and is currently excavated by a team led by the University of Ferrara. Human occupation is attested by numerous lithic artifacts and combustion features, and the site has produced unusual finds such as ornamental objects and evidence for intentional removal of bird feathers. The cave is part of several cavities and has accumulated sedimentary sequence, including Mousterian, so layer A11 to A4, the traditional industry, the Ulusian, which is layer A3, and the Aurignacian, layer A2 to D3. Several studies provide dates placing layers A4, so assigned to final Mousterian, and A3, which is Ulusian, between 41.3 and 39.1 Ka. The bone assemblage is highly fragmented across the stratigraphy, with only 3% identifiable based on morphological evidences. Here we can see the dominant faunal component for both layers A3 and A4, which is mostly red deer and ibex, and the difference occurs in the abundance of these two species. So 684 morphologically unidentifiable bone and dental specimens have been randomly sampled across layers A6 to A2, with 73% deriving from layers A3 and A4 and 98% of successful identification rate. Taphonomic informations have been recorded on all identified and unidentified specimens by experienced zooarchaeologists, providing consistency and comparative datasets between ZooMS and morphology components. Our comparison of species abundance between ZooMS and morphology component contrasts with previous studies. Indeed, previous non-targeted ZooMS studies at the sites of Les Côtés, Grotte du Renne, Quincé, and Pinhole Cave have shown that ZooMS and morphologically identified components from the same layers are comparable in both species and abundance. However, at Fumane, while species composition is similar, abundance varies between both components. So there is a six-fold increase in the number of both bison specimens in the ZooMS component, and such frequency difference in the presence of a particular species between ZooMS and morphology component of the same archaeological unit has never been observed. So then we have decided to investigate the causes for this compositional difference. So we focus on the ZooMS component and the three main taxons, so Capra here in green, Cervids in dark blue, and Boss bison in light blue. First, we looked at the primary potential cause for this compositional difference, which is bone fragmentation. So bone fragments show here no significant difference in overall bone length distribution, as illustrated by the figure on the left, and there is no apparent spatial patterning when these fragments are plotted onto the excavated area, as shown by the figure on the right. So we've just seen that studied bone fragments have nearly identical distribution of specimen length. Assuming that the Boss bison individuals are generally larger than Capra and Cervids, Boss bison bone elements have been thus subjected to a larger amount of fragmentation. Secondly, we looked at molecular diagenesis, more specifically, an indicator of preservation of collagen through peptide deamidation. Deamidation is the conversion of the amino acids glutamine and asparagine into respectively glutamic acid and aspargic acid. This modification may alter the structure and stability of the molecule and can lead to protein degradation. We observe that there is no significant relationship between bone length and deamidation values across the species group, as shown by the figure on the left. There is no special difference of the average deamidation per square, as shown by the figure in the center. And also, we find a similar distribution of molecular diagenesis between layers A3 and A4, as shown by the figure on the right. So deamidation cannot explain what we see in terms of species proportion. When considering taphonomic processes that could have caused such fragmentation, we notice that the carnivore modifications are limited. However, there is a high abundance of human modifications such as cut marks, filleting marks, and deliberated bone fractures for marrow extraction. This last pattern is shown here, illustrated by percussion marks. So the proportion of surface modification for the morphologically identified component is similar between species. Percussion marks are absent within Capra and Boss bison and occur at 3% within Cervids. However, the proportion of anthropogenic modification are more distinct within the ZooMS component, with the high frequencies of percussion marks for both bison specimen, with a mean of 24% for both layers A3 and A4 combined, while it occurs at much lower frequencies in cervids, so 2 to 4%, and is absent within capra. Such increased fragmentation resulted from a specific hominid behavior during food procurement, providing a potential behavioral explanation for the six-fold increase in both bison remains. While the morphologically identified component of a faunal assemblage may be dominated by a single species, this may not necessarily reflect true assemblage abundance. Without ZooMS, both bison were previously considered a minor component of the Fumane fauna. But the larger size of the bone elements, the higher frequency of long bone shaft, or the marrow fractures in the ZooMS component could explain the higher incidence of this species. We've seen here with these two case studies that integrating complementary data sets from the archaeological and ZooMS-based analysis in a systematic manner provides a more accurate picture of species proportion and highlight variation in the intensity and treatment of different prey sizes. I would like to thank all the persons who contributed to these projects, but also thanks to the organizers for welcoming me here, and thank you all for your attention.